Good afternoon, everyone. Thank you for hanging on until now. So I'm Shakil Hosain. I'm a PhD candidate in plant ecology and microbiology department at Texas A&M University, which is this department, and I work with Dr. Jim Shaw. So a little bit background, as you have heard before the break, and then we recently published our transcriptome data set where we reported that um, the Rosette virus accumulates significantly in symptomatic branch than asymptomatic branches. And we hypothesized that maybe the differentially expressed gene could lead us a, a mechanism of resistance that might be causing the asymptomatic branch as asymptomatic. That means no symptom, even though um, even though it's infected, but show no symptom. So some of the questions that catalyzed our current research are okay. So down there you see a healthy plant which is not infected, and you see a infected plant. We show two different phenotypes. One side shows symptoms, the other side doesn't show any symptoms. So out in the field, when readers are scoring visually or visual inspections, it might be misleading, saying that okay, maybe this is a healthy plant, but in reality, it is actually infected plant. So when it comes to resistance in terms of phenotypic resistance out in the field, the no the absence of Rosette symptoms might be an indicator of phenotypic resistance. So few of the question that we are wondering, does the asymptomatic regions of the infected plants represent resistance? Maybe a systemic acquired resistance for SARS. We're also wondering, how does virus infections alter the hormone balance? And finally, we are also wondering, can the transcriptomic differentially expressed gene reveals defense mechanism? Okay, so we profile 44 metabolites with LCMS profiling. And here we are focusing on two major hormones, salicylic acid and jasmonic acid. This is a very busy slide, so I will try to break it down for you. Let's look at a free jasmonic acid. You can clearly see the level of jasmonic acid alter from healthy plants to infected plants. So these two panels are infected, the middle being the asymptomatic and the right one being symptomatic. If you look at the bioactive form of the jasmonic acid, which is the group down here, which is the amino acid conjugates of the jasmonic acid, you can also see the altered patterns of this jasmonic acid. So one of the most common bioactive form of jasmonic acid is the jasmonic acid isoleucine. You can clearly see the level of the bioactive form of jasmonic acid are altered within the infected plants. More specifically, their difference in asymptomatic branch or asymptomatic cane from the symptomatic cane. If you look at more closely, the conversion of free JA to the bioactive form of JA, this is also altered in these two forms. You see from here, free JA is less here, but when it's converting to bioactive forms, there are more in this asymptomatic plant. Overalls, their levels are low in the symptomatic branch. If you look at the inactive form of the jasmonic acid, which are 12 carboxylate jasmonic acid isoleucine and 12 hydroxy jasmonic acid isoleucines, you can also see an alter patterns how they're accumulating in healthy plants than in infected plants. So might be telling us that J defense contribute to the limiting Rosette virus, because we have shown that the accumulations of this virus is less in asymptomatic can than in symptomatic can. We also looked at the 13 LOX metabolites, which lead the Jacqueminot biosynthesis. Here, if you see all the 20 13 LOX metabolites, you can also see the altered pattern of accumulations in infected canes than healthy canes. Here you see a diversion in the pathway of the jasmonic acid biosynthesis, which is the alpha-linolenic acid pathway. So there are diversion happening. So all these metabolites are increased in asymptomatic cane, but in the middle you see these three metabolites are accumulating more in symptomatic cane. Let's look at another major phytohormone, which is salicylic acid, that confer resistance against biotrophic pathogens. If you look at the level of salicylic acid, you can clearly see the salicylic acid's pathway is also altered in infected cane than healthy plant. Let's look at two of the metabolites that upstreams of phenylpropanoid pathway that leads the salicylic acid biosynthesis. So coumaric acid and cinnamic acid, these two sits upstreams of the phenylpropanoid pathway that later convert to salicylic acid. Here you can also see the clear alters of these two metabolites in infected plants than healthy plants. If you look at more closely in asymptomatic canes, these two metabolites are relatively accumulating more. So since they're in the same pathway, and these are the upstreams of this phenylpropanoid pathway, we can assume that the salicylic acid accumulation should be more in symptomatic as well, because they later convert. They're in the same pathway, they're upstream, so downstream they should be more. But here you can see a clear block of the salicylic acid accumulations in the asymptomatic branch. So, okay, we talked about it. We also looked at 9 LOX metabolites, which is also in the alpha-linolenic acid pathway. 9 LOX metabolites are known for insect herbivory and the cell wall defense. Especially if you look at azelaic acid prime plan for salicylic acid mediated systemic acro resistance. Here you see among all these 9 LOX metabolites, you see it is accumulating more in asymptomatic. We talked about salicylic acid in last slide, and we are saying that azelaic acid prime plan for salicylic acid mediated systemic acro resistant. So in the asymptomatic branch, the reason it is showing less symptom or no symptoms, it might be systemic acro resistance is causing the branch to be symptomless. And then we wanted to look at what is happening in the biosynthetic genes when it comes to differentially expressed in our transcriptomic datasets. We find that the virus infection or Rosette virus infection alter lipid-derived signaling, alters calcium signaling, and also assimilated defense. How we saw that, we looked at hallmark genes for systemic acro resistance. For instance, TGA10 and PR1, PR genes, we see their differentiating patterns upon Rosette virus infection. We also see, when it comes to lipid-derived signaling, we also saw alanine oxide synthase, PIPLC, phospholipase C, 13 log, they are differentially expressed upon Rosette virus infections. When we are talking about calcium, calcium signaling also altered upon Rosette infection. We saw genes that are involved in calcium signaling, for example, chromosomal binding gene. If we look at diagrammatically, where in the pathway those genes are altered or differentially expressed on Rosette virus, these diagrams show clearly that in the plasma membrane localized phosphoinositol phospholipase C, it gets altered, which release triphosphate and diacylglycerols, which later activate the calcium response. We see the gene that are differentially expressed, they get targeted here as well. We see the oxylipins and jasmonic acid biosynthesis in the chloroplast from unsaturated fatty acid. These genes are getting differentially expressed upon Rosette virus infections. We also see the SCRI, which is salicylic acid binding protein 2. It converts inactive salicylic acid to an active salicylic acid. So SCRI convert methyl salicylate, which is the inactive form of salicylic acid, to salicylic acid. And that gets recognized by NPR1, which is the master regulator or hallmark of salicylic acid mediated systemic effect resistance. It gets targeted here and differentially regulated here as well. The NPR1s later get perceived by PR gene, which is pathogen resistance gene, which confer immunity against pathogens. And we see differential express of pathogen-related genes getting targeted here as well. We saw azelaic acid. We talked about azelaic acid that is related to the DNA methylation and his modifications. We also talked about PR genes, how they are getting targeted here. So overall, we see upon RRB infections, signal transduction pathway is getting affected. A phosphorylation pathway is getting affected, and hormone biosynthesis are altered as well. So now we wanted to look at more broadly of the stress and defense responses spectrum that are present in our transcriptome data set, the genes that are differentially expressed. We wanted to look at them broadly in terms of stress and defense responses. So we limit the gene ontology terminology by level 3, and these are the terms we find that are present in our transcriptome data set. Even though we did not see a difference in terms of the terms or category or group, however, we see the number of genes, they are actually different across the category and tissue as well. You can see here the response to chemicals, the number of genes, they are actually different in each category. If you look at the Venn diagram, you can clearly see that actually there are no, they are unique genes. They are not shared. So these genes, if you map them in the genome, you can see their distribution is across the genome. We'll be focusing on chromosome 5. In today's talks, we have heard chromosome 5 a lot. In Black Spot as well, 2 talks, they're focusing on chromosome 5. We'll talk chromosome 5 in this talk as well. We saw previously that upon RRB infections, the signal transduction pathway, phosphorylations, and hormone signal is getting affected. So signal transduction pathway, which are broadly are kinases. So we wanted to look at the molecular kinases that are present in our data set and that are differentially regulated or differentially altered upon RRB infections. So these are the number of kinases. If you look at the Venn diagram, you can also see that they are not common; they're actually unique kinases, which are differentially expressed upon Rosette virus infection. So most of these kinases are protein serine threading kinase, which are typically linked to defense responses. So after that we wanted to look at the resistance gene in our data set. We talked about like molecular kinases, protein serine kinases, which are linked to disease response that typically happens to be a disease resistance gene. So we're going to predict the resistance gene that are present in our data set. To predict that, we use disease resistance analysis and gene ontology, which is called Drago pipelines, in the plant resistance gene database. This is an interface of the database. So these databases predict the genes based on their predict the resistant gene based on their domain architecture. More specifically, based on the disease resistant genes domain architecture. So our analysis revealed that in our transcriptomic dataset upon Rosette virus infection, a total of 228 resistance genes are differentially regulated. If you look at the Venn diagram here, there are 5 genes that are differentially altered and common between symptomatic upregulated and asymptomatic downregulated. So this bar chart shows the distributions of this resistant gene across 4 conditions. You can see there are majority of them fall in symptomatic upregulations and so on. This chart over here, table over here, show the categories of those resistance genes and how they are distributed among the 4 category or 4 conditions. Okay. Now we wanted to look at linkage group 5. Why? Because Dr. Oscar Schwinger's group focused on a genome-wide association study and QDL study, and throughout their multiple-year experiment and study, they identified or they reported that QDLs in linkage group 5 heavily related to Rosette disease. Actually, 40% of the phenotypic variation is explained by these QDLs who are resides in linkage group 5. So they reported 3 meta QDL. Well, I don't know why the slide a little bit cut down here, so I cited their work there as well, if Dr. Oscar is wondering. So they reported in 2023, they reported 3 meta QDLs that are explaining 40% of the kind of variations in disease severity for Rosette disease. So if you look at this chromosome, if you look at the this QDL, this is the length of this QDL, and meta QDL, meta RD3 is the biggest one or the largest one, which comprises 20.81 megabase per. So we wanted to do a comparative analysis among the differential express gene and the gene reside into those QDL in linkage group 5 to see how many of those genes in linkage group 5 in those QDL are differentially expressed than we found in our transcriptome analysis. So I extract all the genes that are underlined in those meta QDL. I found in total there are more than 3,000 genes that are reside in those 3 meta QDL in linkage group 5, and these are their respective number. So this is the total represents. If you see 3, we saw it's approximately 2,000 genes that are residing in QDL and meta QDDR. RD 5.3, which I will refer to 3, 2, 1. So QDL 3 itself is a big and large. That's why there's a more number of genes that are reside underneath this. two is 482, and one is like around 700 of those genes. If you look at the Venn diagram here of, and we found that 141 genes among these are differentially expressed. So our comparative analysis found that there are 141 genes that are differentially expressed that are also reside in this region. And if you map these 141 gene in a Venn diagram, you would also see they are not actually common. Their distributions are unique across conditions as well. And this heat map shows they are differentially expressed from the RNA-seq or the transcription analysis. So these are 23 genes over here. So they are being curated, they've been sorted from the transcriptive analysis, from the QDL analysis, and from the resistant gene that I showed prediction of the resistant gene. So 3 level of sortings happens to find out these 23 genes, that they are differentially expressed, they are resistance genes, they are in the QDL regions of linkage group Y. And one of the genes that I showed in the previous slide as a 5 genes that are differentially altered. One of them is represented here, is differential expression profile here. It is less in asymptomatic, it is induced more in asymptomatic, which is this gene down here. So we did promoter analysis of those differential express antiviral genes to see what are the cis regulatory elements that are controlling those genes. As you saw before from our lab, from the previous study, majority of those genes actually controlled by the gibberellic acid. Not surprisingly, we also see these differential genes, majority of the cis regulatory elements actually are controlled by the gibberellic acid. We see some of them as a oxine, but when you go up here, surprisingly, we see actually none of them actually regulate the GA. So we wanted to picturize them, how the defense and developments happening or going on upon Rosette virus infection. So this model shows that cell wall remodeling by Rosette virus. So we have receptor-like kinase, which is peronia. So we have 2 peronia-like gene that are differentially expressed in our data set. So they activate the polygalactoranases, and these polygalactoranases release the oligogalacturonidase, which act as a dump, which is perceived by wall-associated kinase, and then they later trigger the pathogens-related gene. We also the phytohormone SA and JA, they actually trigger the Werkes transcription factor, which later can trigger the PR genes. As well, we also find a cyclin-dependent protein kinases, which are differentially regulated, which control the cell cycle by differential JA and ADA response. So broadly, they are more of a trade-off of effective immune systems and development happening, or how the host is balancing symptom development and immunity when it comes to the asymptomatic case. So in summary, we saw these hormone disruptions are different in Rosette and asymptomatic tissues. Antiviral defense restrict Rosette virus infection in asymptomatic cases, but are less effective in Rosette tissue. Virus infection, we saw how virus infection reprogram lipid derived signaling. We also see high level of resistant genes expressions upon Rosette virus infection, and QTLs on linkage group might have key genes which might regulate the Rosette disease. Virus infect plants can show different symptoms of the same type. With that, I would like to thank our lab, Reagan's, my advisor, Dr. Vishwa, and all the collaborators, Dr. Benura from Sri Lanka. He helped us with our bioinformatics pipelines and mentoring as well. With that, I would like to thank you, and I'm open to take questions.